Good morning, everyone, and welcome to this last plenary lecture of the Toronto ACA meeting. And this morning I have the distinct pleasure to award the Martin J. Buerger Award to Michael James. Let me read the citation. This is the Martin J. Buerger Award to Michael James for outstanding macromolecular crystallographer who has made many seminal contributions to structural molecular biology, including the determination of the structure-function relationships of proteolytic enzymes, muscle filament, viral proteins, and antiviral agents as landmark achievements in the structural biology field.
First of all, let me thank everyone for coming on the last day at 8 o'clock and after the wonderful banquet that we had last night. I'm very impressed. I want to thank the ACA through the president and members of the ACA committee for the selection for this award. I'm deeply honored. I can't really find the words to express how much I feel to be honored by my peers and colleagues in this award. I put up the photograph of Martin Buerger for a number of reasons. He was an amazing crystallographer. Any of you who have taken elementary crystallography courses will have recognized his seminal contribution in the writing of 5 very detailed, very explicit textbooks in the field. His elementary crystallography is by no means elementary. And he also developed the precession camera. This is a tremendous achievement, and wrote an extremely edifying book on precession photography. I met Martin in 1964. I was fortunate to be able to go to a NATO summer school in crystallography that was held on the island of Zakynthos. It's just off the west coast of Greece, and I drove a mini minor all the way from Oxford, where I had just started becoming a doctoral student, and it was a terrific trip. It took me 5 weeks. The total trip was 5 weeks long. It was a terrific holiday, and on top of it, I learned all about crystallography. I'm also very indebted to the University of Alberta, who managed to put up with me for 41 years. I've been in the Department of Biochemistry for that long. What I want to do is to just go through a little bit of my history. I've been updated a bit by Ted Baker's marvelous talk on Tuesday, and but I want to give you my perspective of the early days of crystallography and what I've done, and then I'll switch a little bit to the next level, where I'll describe some of the work that we're doing today in my lab. This is Sillimanite, and I didn't know it was a gemstone I worked on something like this, a little piece of that chipped off a sample that my supervisor at the time gave me to work on the structure, and not, again, with Googling Sillimanite just before my lecture, here I come up with all of these beautiful gemstones that are in Southeast Asia, in India and Sri Lanka and Burma. Quite beautiful. Bob Ferguson was my supervisor for my master's degree in the Department of Geology. Bob was an outstanding mineralogist at the University of Manitoba, and we came up, this is not a figure from anything that I've done, but Sillimanite has got 2 aluminums, a silicon, and 5 oxygens in the asymmetric unit, Al2SiO5, and the aluminum in an octahedral coordination is found at these positions, and then aluminum and silicon is shared in these tetrahedra that are sharing a common vertex in the structure. Just writing up my thesis, this is a lesson to you younger people. Writing up my thesis, Charles Burnham, who was also at Storrs, I believe, published a paper in the Zeitschrift for Crystallography that had the whole structure laid out, fully refined, etc. And I had done the structure determination by 2-dimensional Fourier difference maps and refined it in that way. But not to be upset, I pressed on, and I was accepted by Dorothy Hodgkin in Oxford as a graduate student. And the first picture, this is not Dorothy, of course, this is J.D. Burnell. As Ted described the sage who was at Cambridge, Dorothy went to Cambridge to do her PhD. She was a part 1 and part 2 student in Oxford, and she did her PhD under the direction of J.D. Burnell, and they published the seminal paper on pepsin in 1933, in which they had encapsulated the small pepsin crystals within the mother liquor and then taken the diffraction pattern, which showed that in reality it was possible to determine the atomic structure of pepsin in the same way people had been doing structures of small molecules. This is a picture of Dorothy. She's looking at a model of vitamin B12 here, and I particularly like this picture taken in 1969. One can imagine that she's thinking about, as she always thought about, structures, and she's thinking about the structure of insulin that was just determined in that year. So when I went to work with her, I was assigned a series of peptide antibiotics. I didn't work on the insulin project, much later to my, I wish I had, but at any rate, I worked on this compound here that was a bromoanilide derivative of micrococcinic acid, which has turned out to be an antibiotic that's used in animal feed. But the structure and the distribution of these thiazole rings was unknown at the time. But we had these 2 bromines per molecule. Turns out that from the measurement of the density and the unit cell dimensions, you could calculate that the asymmetric unit was 1800 Dalton, and that implied that there were two molecules of this plus anisole from which it was crystallized. A single molecule of anisole is what I was looking for, and I was very fortunate to be able to solve this structure. I dropped the crystal. There was only one crystal, and I dropped it on the floor shortly after taking up this project. And I spent an afternoon on the floor with a little light shining and looking at the reflection to try and find this crystal. Unfortunately, my eyesight was much better than it is now, and I found it, and this was the result. These are the two molecules in the asymmetric unit. This is anisole, the molecule of salvation, and we identified the details of how the thiazole rings were arranged in this particular compound. That was really a great time. During the course of my doctoral studies with Dorothy, in November in 1965, David Phillips announced the determination of the structure of lysozyme, the first enzyme and the third protein structure to be determined ever. And I was fortunate. We went out, my colleagues and I traveled by train up to London, where we attended at the Royal Institution this marvelous display by David Phillips and his colleagues here, Gareth Mayer, Colin Blake, Tony North, and Sarma, with Louise Johnson, the main people that had worked on the structure of lysozyme at that time. It was particularly impressive. Here, they strung from the ceiling a Kendrew model of lysozyme in just the amino acid sequence, the linear sequence, and then to contrast it, here was how it folded up, and they had little lights that came on for the acidic residues, and little blue lights that came on for the base. It was really marvelous display, and you can see all the people. They're all dressed in black tie, and it was a very formal event, and lots of fun to be there. I was somewhere over here, approximately where the photo was taken, way up in the gods, watching this. This was the seminal event that turned me on to doing protein crystallography. I said, that's what I want to do, and subsequently to that, we've done it. I have to thank Louise for this photograph and this distribution here that she kindly gave me for this talk. Here's a picture of Max Perutz and Tony North and Bob Diamond, who was the person that wrote all of the computer programs for the Ferranti Mercury that we used for calculating electron density maps and doing refinements and so on at Oxford. And here's Louise talking to Helen, Susan Lowy. And it's wonderful here to read Max's comments here. Certain moments are deeply engraved in my memory. One is the Monday morning in March 1953 when Crick called me into his room to show me his and Jim Watson's double helical model of DNA, which immediately revealed the molecular basis for heredity. And another is the moment when David Phillips, Louise Johnson, and Charles Vernon made me first understand how an enzyme works. And these are marvelous words and a tribute to the work of David Phillips at the Royal Institution. So I went back to Canada, to the University of Alberta, and I was very kindly offered a job after a postdoctoral fellowship in the chemistry department. I was offered a job in the biochemistry department to help them set up a protein crystallography lab. And I remember one Friday afternoon, my colleague Larry Smiley brought me down a sample of a proteolytic enzyme and said, "Well, here's SGPB. It's a bacterial protease, protease B from Streptomyces griseus." And within the afternoon, I personally, it's about the only time I've ever grown a protein crystal, came up with these beautiful little cubic habit and crystals of SGPB. And we measured the unit cell dimension. I said, oh my goodness, it's got 153 angstrom cell dimension. I can't work on that as the first protein. So we changed the pH. This was at pH 7, and we changed the pH to 4. 4.5, and these orthorhombic crystals came out, P21212. And although you can't see these diffraction, they were absolutely gorgeous. They extended at the time on these precession photographs to about 2.8 angstroms. So that's what I started on, and here's the first map. And this was a structure in which Penny Cotting and Louis Delbert all assisted in working on. This is the phasing information that you get, and this was the mean figure of merit as we go out in resolution here. So it was a very well-determined structure from two heavy atom derivatives, and you can see from stacking up the electron density, sheets of electron density parallel to the c-axis, you can see the solvent channels between the molecules, and man, that was really exciting. But sorry, this is not 2.8 angstroms. This is 4.5 angstroms. I'm sorry. But so then we extended it. And Louis Delbert, who everyone here I'm sure knows, and was a tremendous tour de force in extending this resolution to 2.8 angstroms, in which we could actually see the residues, the histidine in behind it, the aspartic acid, and the serine here with a phosphate bound in the active site. And these were really exciting times. The molecule, this is how we fit the electron density with a Richards box. This is Yinan Xu, who's working on another enzyme, penicillopepsin. And we all had mustaches. Well, Yinan didn't, but we had mustaches and sideburns and lots more hair than I have right now. This was a very exciting project that was developed with my colleague here in the University of Toronto, Theo Hoffman, and we determined the three-dimensional structure of it. And it was great. Yinan went on after his postdoc period in my lab to California, where he, I think he's still there, but I'm not absolutely certain. So this, you needed to have short people like Yinan to be able to build these models, and it was great. I had my technician Koto Hayakawa, who with Yinan 123 amino acids, they put the whole thing together in this very cramped space at the time. Going back now to SGPB, this is the drawing that was in the paper that we published on the structure of SGPB. And then subsequently, Ting-Wei Li has been working on a number of different complexes of SGPB, and this is what the molecule looked like in 2007. Of course, it's much prettier, but if you look, it has exactly the same fold that it had in 1975, which is a good thing. And I then established a wonderful collaboration with Michael Laskowski Jr., perhaps one of the best protein physical chemists, certainly that I ever came in contact with. His major project was working on the structure of how molecules associated together, and what determined the association forces between these molecules. And so we were able to assist in this project by working on the third domain of the ovomucoid, which is a three-domain molecule, from turkey eggs. And so this is the third domain here, and between this leucine and glutamic acid is where serine proteases would cleave, were this not a very good inhibitor. It's a very, very bad substrate, but an excellent inhibitor. It takes any of the proteolytic enzymes approximately 24 to 36 hours to cleave that bond. So we his plan was to choose a family of proteins, and he selected the casal inhibitors, this family of the third domain, and the wild type that he chose was the ovomucoid from Turkey. And then there was a series of targets. He had 6 targets: chymotrypsin, porcine pancreatic elastase, human leukocyte elastase, sublysine Carlsberg, and SGPA and SGPB were the 6 targets. And what he did was to purify not just the wild type from Turkey, but in determining the three-dimensional structure of the complex, we could identify that there were 10, colored blue here, 10 residues that made intimate contact with these serine proteases in these complexes. And what we did was then, what he did was to change each one of these to all 19 amino acids, all 20 coded amino acids in turn, and measure very, very carefully the association constants between them, and then come up with this algorithm whereby he was able to predict from the sequence the binding constants for these molecules. So this is the structure of OMTKY3, and here's the little segment of polypeptide that fits into the active site of the protease. The P1 and P1 primed residue, the leucine is pointing into the board, and the glutamic acid is coming out of the board, and you can see that involving another residue, asparagine 33, it's highly hydrogen bonded in this region, and then there's 3 disulfide bridges, one here, here, and here, which give this molecule an incredible amount of stability. And it was the additivity by changing each amino acid in turn and looking and comparing it to the wild type that gave him this idea for his sequence to binding constant algorithm. So here's 6 of the amino acids, 6 of the 10 amino acids that are binding in the active site as they meander through. Here's the P1 residue is a leucine in the wild type enzyme. P1 primed is a glutamic acid which hydrogen bonds to this threonine in the P2 position. The disulfide bridge at cysteine in the P3 position, an alanine and tyrosine and an arginine. So you can see how beautifully it fits into the active site, and even if there's room left, you get some water molecules that fill up the extra space. Here's this leucine in the P1 position, the carbonyl oxygen in the oxyanion hole between the nitrogen of serine 195 and glycine, the NH of glycine 193, and the hydrogen bond between the serine and the histidine, and the hydrogen bond from the histidine to the aspartic acid, which is there in the back. And it fits absolutely beautifully in this active site. And then we went on to look at additivity and non-additivity. Here's an example of additivity with SGPB, changing the P2 residue and the P1 residue, which is a threonine and a leucine, from threonine and leucine to lysine and arginine in the P1 position. Of course, it binds much less tightly in terms of the delta G values, about 5 kilocalories per mole less. But if you look at the diagonal segments of the delta G here, they're almost identical. Here, changing the P2 from a threonine to a lysine, it goes down by 2 kilocalories, and similarly, changing, you've already changed here now the P1 to an arginine, changing the P2 to a threonine, it also goes down by 2 kilocalories. So the delta G between these values is only 0.1 kcals per mole. However, this is considered additive. On the other hand, with chymotrypsin, if you change the P2 residue threonine and the P1 primed glutamic acid, this delta G value is enormous. It's almost 3 kilocalories per mole, and these diagonal terms don't agree. So this is an example of non-additivity, but by changing the threonine and the glutamic acid individually. So another example was glycine 32, and alanine was changed to an alanine 32. And the difference, the delta G between these two was about 1.3 kcals per mole. The alanine binds not so badly, but it certainly is a weaker binding, but only by this 1.3. But it is a non-additive. It is non-additive. And so Ting-Wei Li determined the three-dimensional structure of the alanine 32 and compared it very carefully with the wild-type glycine 32 here in the blue color, and the alanine is in this mauve color. You can see the enzyme essentially doesn't move. It fits extremely well, but the inhibitor shifts from here to here. And Ting-Wei was able to identify why that happens. And here's glycine 32, and it normally makes a contact with this proline 192B in SGPB, the alpha carbon of glycine 32. But now if you put a beta carbon on it, there's no room for that beta carbon here. So just putting that methyl group shifts the whole molecule through this about a 6-degree rotation and a subsequent weakening of the association constant by about 1.3 kcals per mole. Well, again, another small change was examined, and that was the loss of one hydrogen bond by changing the amide from the P2 to the P1 position. P2 again here is threonine, P1 is leucine. This is the pocket in which the P1 residue binds, changing it from an amide to an ester. And in the native structure, in the wild-type structure, that NH forms a hydrogen bond to the O-gamma of serine 195 in the back there, which also has formed a hydrogen bond to histidine 57. And it's also another hydrogen bond to the carbonyl oxygen of residue 214 down here. And if we change it to an ester, now it doesn't have the hydrogen to donate to these two amino acids. And it is associated with a reduction in the association constant of 1.5 kcals per mole. And the only difference between these two structures, done very nicely by Kathy Bateman for her PhD degree, is the loss of this one hydrogen bond, and we can clearly associate the energy associated with that hydrogen bond to 1.5 kcals, because there are no other differences in the molecule. So that's been two examples of dramatically different effects with a relatively small Emil, how am I doing? Am I all right? Okay. He gave me a serious warning, so I'm really worried about what's going to happen if I go overtime. I'd like to switch to a modern or a relatively recent topic in my lab at the present, and that's we're looking at a variety of different molecules that are made by the Mycobacterium tuberculosis. And so what is tuberculosis? An infectious airborne disease that's caused by inhaling inhaling droplets that contain this bacteria. And if you're riding on a bus and your neighbor is infected by TB and coughs, you're going to breathe this in. I mean, it's inevitable. And if you have a cough that's lasting for more than 3 weeks associated with a fever, you should have a serious medical examination and a chest x-ray. The fact is TB is the leading cause of human and adult death caused by bacteria. There are 2 billion people in the world today that are affected by TB. That's 1 in 3. So you just look at your neighbors and one of you. 2 million people die every year from this horrific disease, and it's mainly children on the African continent. It's becoming much more resistant to treatment by effective antibiotics, and there is a multi-antibiotic or multi-resistant strain. There's an extremely resistant strain for which there are no drugs, and both of these are increasing around the world. Affecting mainly the lungs, and you can identify regions in the lung that have these little tubercles where the organism is living inside macrophages that are designed. Macrophages are part of our immune system, and they're designed to kill these organisms, but TB has learned how to live and as a latent infection in our lungs. They have been found as long ago as 5,000 years ago in lung tissue of Egyptian mummies. The Bronte sisters, 2 of them died from TB. Dostoevsky had TB. Chopin had TB. Why is it? I mean, it seems that it's all of these wonderful arts people that had these terrible diseases. John Keats had TB. Edvard Munch, with the artist that drew The Scream. Franz Kafka. And more to our day, Tom Jones had TB when he was 14 years old. Vivian Leigh from Gone with the Wind. She died in 1962 from TB. And the physicist Richard Feynman had TB. And the man that discovered TB is Robert Koch, who discovered it in the mid-1850s. So it's a prevalent disease and still is existing in our day. So we've joined the Structural Genomics Consortium on TB that's run now currently from Texas A&M with Jim Saccatini. Formerly it was at Los Alamos with Tom Terwilliger. And we've targeted a number of molecules and enzymes from the TB. And from that, currently we've made about 500 primary clones and sequence confirmed 450 expression clones, developed preliminary expression vectors for 425 of those 450, and expressed about half of those on large scale. And we have 90 of those proteins that have been expressed that are either currently in crystal screens or have been crystallized. Successfully, 50 of them have been crystallized. We've refined, determined the structures and refined 39 of them, and 26 are currently deposited in the PDB. This is as of July this month. I put down in particular Craig Guerin, who's my head molecular biologist, protein purification. Craig is just an outstanding person. And also Maya Cherney, who's been involved in the crystallization of a vast number of these these molecules. Maya has been in my lab since the early 1990s and has been wonderful in doing the crystallizations. So these are some of the molecules that we've determined. I think if you went to the poster session on Tuesday, Maya had the presentation on RV 1657, which is the arginine repressor, and it's a complex with DNA, and there's a little bit of and arginine. So it's really a very interesting structure. 1653, Sankar had a poster on either Sunday or Monday showing this structure. And I think that's all that we had posters for. No, it's not all. Jiang Yin had a poster on RV 1018, but I didn't put it up here. Okay, so we've targeted a number of biosynthetic pathways, and one of them is the arginine biosynthetic pathway in Mycobacterium, and this just shows you the pathway from glutamate to arginine, and we've done the structure of 1652. Here's some beautiful crystals. And what I want to talk about is the structure of RV 1656, which is an ornithine carbamoyl transferase. So it's a relatively simple reaction. Of course, the ornithine in a solution by itself would have a positive charge on that amino group, but in the enzyme, it's probably lost the proton and it's neutral, so that it can act as a as a nucleophile to attack the carbonyl carbon of carbamyl phosphate here to give you citrulline, which is the next product on the reaction pathway. And this is the expression, gateway cloning. We amplified the gene by PCR, vectors here, expressed in E. coli, BL21, P. lyse cells, incubated until the optical density was about 0.8, and then induced the production of RV 1656 by the addition of millimolar IPTG, and it was purified by a nickel affinity column, and you can see that it gives a very beautiful protein solution. And we were able to grow, Maya was able to grow two different crystal forms, a hexagonal crystal form here under those conditions, and not terribly different conditions produced this orthorhombic crystal form. So Sankar collected data from both of these. The orthorhombic is P2121, and the hexagonal was P6522. And you notice that there's a 463 angstrom axis. So that was a bit challenging because we had to move the crystal to detector distance back at the synchrotron to about 500 millimeters in order to collect the data, or to have the data individually separated in the diffraction pattern. But everything went swimmingly well. R-sim for the R-sim for the orthorhombic form was a bit high. R-sim for the hexagonal form was really nice, but we didn't get 100% of the data, unfortunately. We only had this one really nice crystal for the data collection, but it refined beautifully. Both of them did, and they're nice structures. So then it's a trimer. Ornithine Transcarbamoylase is a trimer, and it's reminiscent of aspartate transcarbamylase, the very famous structure that Bill Lipscomb determined. It doesn't have these regulatory domains, but it has these three catalytic domains that ATC has. There's 6 in ATC, but there's only these 3. So the active site is just in here, and parts of this molecule of the trimer contribute residues to the active site, as you'll see in a minute. Sankar was very lucky to be able to co-crystallize in the hexagonal form norvaline, sorry. It doesn't have the nitrogen in this position here on the side chain, but the amino group and the carboxyl group. Excuse me, carbonyl phosphate is here, and the carbonyl carbon atom that is attacked by the nitrogen in the normal reaction is this one here, with the release of phosphate. So it really gave us the idea of how this enzyme worked. That's just an overview of the whole molecule. And if we zoom in, the phosphate of carbonyl phosphate is stabilized in its position by this the N-terminus of this helix, which would have a coupled positive charge, and then a number of hydrogen bonds that are to these 2 oxygens of the phosphate. And then here's the norvaline, which has also got a huge number of hydrogen bonds associated with it. And just bear with me a little bit. You'll see that the carboxyl group of norvaline has a hydrogen bond here, one from water, one from this NH group of methionine 229, and then another one here from water. So there's 4 coplanar hydrogen bonds to the carboxyl group, indicating that this is very likely negatively charged. And similarly, the NH3 receiving 3 hydrogen bonds to it indicates that it is very likely positively charged. And there's especially from this aspartic acid here to that NH3. And then if you notice, there's a series of positively charged groups, this arginine, this arginine, this arginine over here, another arginine down there. So indicating that this whole region is very likely positively charged, and the charge on the phosphate group is being stabilized by these hydrogen bonds from that helix that we see, that just the first turn of that helix winding around that phosphate group. So it led us to think that, gosh, you can't bring a positive charge on ornithine into this highly positively charged region, and probably it's going to come in here as NH2. Okay, let me quickly go ahead. Here it is, another view of the active site. And our mechanism that we came up with was the ornithine is coming in as an NH2 that would be able to attack the carbonyl phosphate oxygen. There is indeed an oxyanion hole that has hydrogen bonds to that oxygen that will help to stabilize a negative charge that develops on that. And the phosphate is part of the carbonyl phosphate is part of the mechanism, and it would be able to accept a hydrogen from that nitrogen to give you citrulline and phosphate. Another group had done the structure of human OTCase and used as a general base the cysteine residue that is close by, but it's not close enough, unfortunately. And it would have given up its proton to aspartate, the one that's forming this important hydrogen bond to the NH3 of ornithine. We disagreed with that, and I'll show you in the next view why that mechanism is probably untenable. The distance from that sulfur is about 6 angstroms, and you cannot really transfer a proton from that nitrogen to the sulfur. It's very unlikely a cysteine residue with a pK of 8 would transfer a proton to an aspartic acid with a pK of 4 as well. So this summary, we found 2 crystal forms. Carbonyl phosphate and norvaline induce a large amount. I didn't talk about that. Interactions involving carbonyl phosphate and norvaline negate earlier proposals that were based on the human enzyme. Our structure suggests it's a zwitterion with a neutral no net charge on the nitrogen of ornithine, and that phosphate is likely contributing to the mechanism. So let me just thank everybody. I mean, I'm thanking everybody that's contributed to this Buerger Award, and I'm deeply indebted to all of these people for the work. Excuse me, for the work on SGPB and the ovomucoids. Michael Laskowski Jr. At Purdue and all of his students and postdocs, and Stephen Anderson, who developed the expression system for the ovomucoid as well. And this is my current group. I have 2 students, Nobu Watanabe and Mridula. Mridula's project is on prion proteins. Nobu works on lysine biosynthesis. Maya, the crystallizing person. Craig Garen's just hiding in the back there. Sankar is here. Bindu is here, and Jang is here at the meeting. And Kathy Bateman is also a member of my group. And, you know, here you won't be able to read it, but these are all the people that have been in the lab over the years, and I really hope I haven't left anybody's name off. Thank you very much.